Hello, welcome to a section of series of my presentation. I am Samuel Owoeye, popularly known as Exopient. Now, this presentation features blood physiology. But before we proceed, let's have the highlights or outline of the presentation. Number 1, we go with introduction. Number 2, we look at the blood parameters. Number 3, we look at the functions of blood. Number 4, we look at the components of blood. Then number 5, we look at the blood cells, as the red blood cell, anemia and polycythemia, white blood cells and immunity, then platelets and hemostasis. Then finally we look at red blood cells, antigenicity. Now for the introduction. Now blood is a fluidic connective tissue that circulates throughout the body inside a series of connected channels that we call blood vessels. Now blood is actually red in color because of large amount of red blood cells. The red blood cell is red because of hemoglobin, that's a red pigment. Then hemoglobin is red because of heme inside it. Heme is actually red because of iron too. So we can say in actual sense that blood is actually red because of the presence of iron inside. Okay, now let's look at the blood parameters. Now the blood volume is averagely 5 liters. Blood is actually 8% of the body weight, while the blood pH is actually between 7.35 to 7.45. Then viscosity is actually 3 times of water. Now let's look at the functions of blood. The functions of blood are nutritive function, respiratory function, excretory, defensive, storage, transport, and regulatory functions, such as the regulation of acid-base balance, regulation of water balance, and the regulation of body temperature. Okay, now let's look at the components of blood. The components of blood are the blood cells and the plasma. Now the blood cells, they actually originate from pluripotent, hematopoietic stem cells. Those are the uncommitted stem cells, and these will actually give rise to different line of blood cells, being acted upon by different growth factors and differentiation factors. All right, now let's start with the most popular blood cell, which is the red blood cell. Now red blood cell is a non-nucleated biconcave shaped cell, and the normal count for male is 5.2 million, while for female is 4.7 million, plus or minus 300,000. Now the origin and formation of red blood cells, that is the erythropoiesis. Now for the pro-erythroblast, give rise to early normoblasts, which is also the same as a basophilic erythroblast, which give rise to intermediate normoblast, which is also known as polychromatic erythroblast. Intermediate normoblast give rise to late normoblast, which is also known as autochromic erythroblast, which give rise to reticulocytes that actually entered into the blood via diapedesis. Then reticulocytes will now become a mature erythrocyte within 1 or 2 days. Okay, now this erythropoiesis actually covers 5 to 7 days. Okay, and the normal lifespan of a mature red blood cell is actually 120 days. All right, now let's look at the functions of red blood cells. Red blood cell is involved in transport of respiratory gases such as oxygen and CO2. It has a buffering function, that is the presence of hemoglobin in the red blood cell. Then it's also involved in blood group determination, that is the red blood cell antigens, on its surface. Then it's also involved in generation of blood pressure, and also it also causes the blood viscosity. Okay, now red blood cell majorly contain hemoglobin. So let's look at this hemoglobin. Now hemoglobin averagely in a male is 16 gram, while in female is 14 gram per 100 ml of blood. Okay, and hemoglobin actually carries oxygen from lungs to tissues, while it carries CO2 in return. Okay, now hemoglobin has 2 components, the heme and the globin. The heme contains iron 2, while the globin contains amino acids. Okay, now looking at iron is transported in the blood as transferrin and stored as ferritin and hemosiderin, that is in the insoluble form. Okay, now let's look at anemia. anemia is actually decrease in oxygen carrying capacity of the blood, or the deficiency of hemoglobin in the blood, which is actually due to too few red blood cells in the blood or low hemoglobin level in red blood cells. Okay, anemia can be classified etiologically or morphologically. In other words, etiologically, that's based on the cause of the anemia, while morphologically that's based on the form and structure. As when you look at the size, the structure, and the hemoglobin contents of the red blood cell. All right. Now let's also look at the types of anemia. We have a number of types of anemia, like blood loss anemia, iron deficiency anemia, aplastic, megaloblastic, hemolytic, pernicious, sideroblastic, and thalassemia. Alright, let's look at the signs and symptoms of anemia. The common signs and symptoms of anemia can be like jaundice, like cardiac murmur, tachycardia, dyspnea, tachypnea, chills and cheilitis, pallor, fatigue, headache, dizziness, and intermittent claudications. Okay, finally, let's look at the effect of anemia on circulatory system. Now, this eventually leads to acute cardiac failure. Now, it goes like so: decrease in oxygen carrying capacity of the blood, which is also the same as deficiency of hemoglobin, actually causes low blood viscosity, which actually increases the velocity of blood flow, which will increase the venous return. Increase in venous return will actually increase the cardiac output. So at rest, the act is actually exerting the maximum cardiac output. So when the person now go on exercise, now the heart will experience what we call acute cardiac failure. Okay, because there will be a decrease in heart contractility. Okay, now let's also look at polycythemia. Now, polycythemia can be in two forms. You can have secondary polycythemia, which is also known as physiologic polycythemia, and polycythemia vera. Now, secondary polycythemia is due to hypoxic condition, while polycythemia vera is due to genetic aberration. Alright, so that actually ends the section on red blood cell. Now let's look at white blood cells. White blood cells are nucleated mobile units of the body that play important role in body defense system. Okay, they are actually in two forms. We have the granulocytes and the agranulocytes. The granulocytes are the neutrophils, the eosinophils, and the basophils. Right? The agranulocytes are the monocytes, which give rise to macrophages and lymphocytes. The normal count is between 4,000 to 11,000 cubic millimeter. Then the percentage of white blood cells: neutrophils ranges between 50 to 70%, lymphocytes ranges between 20 to 40%, monocytes between 2 to 8%, eosinophils between 2 to 4%, basophils around 0.4%. So you can acronymize it as never let monkey eat banana. That's decreasing the percentage. Alright, then we can look at the genesis of white blood cells. Now we have two lineages. We have myelocytic and lymphocytic lineage. The myelocytic lineage give rise to granulocytes and monocytes, while the lymphocytic lineage give rise to lymphocytes. Okay, now let's look at the properties of white blood cells. Now we have a number of them, like diapedesis. Diapedesis is a squeezing through the capillary wall between the endothelial cells. Margination is the sliding over the capillary wall before it actually enters into the tissue spaces. Okay, diapedesis is actually done by integrins on the wall of neutrophils, while margination is done by selectins. Those are cell adhesion molecules. Then amoeboid movement, that is the random movement in the tissue with changes in the cell shape. Alright, now phagocytosis is actually the engulfing of the invading organisms, while finally we have response to chemotaxis, which is attraction to injured sites by chemokines. That is a chemoattractive effect. Okay, now let's now proceed to immunity. Now immunity is an important aspect of blood physiology, as it has basic medical importance. Now immunity is the ability of the body to resist infection by foreign or invading organisms. Alright, now we have two forms of immunity. We have innate immunity and acquired immunity. Innate immunity is a non-specific form of immunity, and we can look at the mechanism of innate immunity. We have 4 major mechanisms: anatomical mechanism, physiological mechanism, phagocytic mechanism, and inflammatory mechanisms. Alright, which you can acronymize as API, that is APPI. Okay, now starting with anatomical mechanism, that has to do with the skin. That's the skin creates a direct barrier, to bacterial invasion and some other foreign agents. And also you have the mucous membrane, which line the body cavities and sinuses. Okay, that's for anatomical mechanism. Now for physiological mechanism, we can look at different chemical substances in different body fluid and secretions. Saliva and tears contain lysozyme, which attack bacteria. Okay, the gastric juice, acidic secretions, the digestive enzymes in the GIT, vaginal acidic secretions, all these are there to actually attack the invading organisms. Defensins and catalysidins secretion by cells in the respiratory tract. Okay, various chemical substances in the blood, such as the complement protein complex, lysozyme, and some basic polypeptides that attack bacteria and other foreign agents. Okay, now we can look at cells still under physiologic mechanism. We can look at the eosinophils, which attack parasitic organisms in the GIT and other body locations. We also have the natural killer lymphocytes that destroy foreign agents, tumor cells, and infected cells. Okay, now for phagocytic mechanism, we can look at collection of phagocytes. Okay, majorly the neutrophils and the macrophages. Alright, now it involves the engulfment of all sorts of bacteria and foreign organisms. Now we can look at the reticuloendothelial system under phagocytic mechanism, or the monocyte macrophage system. Now it consists of monocytes, mobile and fixed tissue macrophages, and also specialized endothelial cells. Alright, they have name in different locations. You can have tissue macrophages in the skin and subcutaneous tissues. You can have lymph nodes macrophages. You can have alveolar macrophages in the lungs. You can have Kupffer cells in the liver sinusoids. You can have spleen and bone marrow macrophages. You can have microglia in the brain. You can have osteoclasts in the bone. That's it's involved in bone resorption, so it's phagocytosis. Then you have Peyer's patches in the distal small intestine. Then finally, let's look at the last mechanism for innate immunity. That's inflammatory mechanism. Now it actually involves the walling off effect of inflammatory reactions. Okay, now inflammation is the response of injured body tissues to various noxious stimuli, okay, which can be caused by bacteria, trauma, chemicals, heat, and so on. The chemical substance involved in inflammatory reactions are histamine, bradykinin, serotonin, prostaglandins, and some products of complement system. Alright, now let's look at acquired immunity. Acquired immunity is an extremely powerful specific form of immunity, which is also called adaptive immunity. It's a function of lymphocytes. Okay, now the types are the humoral and the humoral or B cell immunity. The types are the humoral and B cell immunity, and also cell mediated or T cell immunity. Now, preprocessing of lymphocytes. Thymus gland preprocesses T cells, while liver and bone marrow preprocesses B cells. Preprocessing period ranges between the fetal life and after birth. Okay, then let's look at the importance of preprocessing. Now it actually helps in the creation of body immune tolerance, the specificity to different foreign antigens, and growth, maturation, and differentiation of B and T lymphocytes in those preprocessing sites. Alright, now clone of lymphocytes. The clone of lymphocyte is a collection or aggregation of genetically identical lymphocytes which are specific to only one antigen. Alright, now formation of memory cells. Memory cells can be in two forms. You can have B and T memory cells. Now for memory B cells, now B cells are activated by a specific antigen. Then activated B cell give rise to lymphoblasts. Now some lymphoblasts give rise to plasmablasts, which give rise to plasma cells. Then that one secretes antibodies. But few of the lymphoblasts that do not form plasma cells actually give rise to new B cells that are genetically similar to the original clone of B cells. Okay, now the collection of the original B cells that was activated by a specific antigen and a newly formed B cells actually formed what we call the memory B cells. All right, now they actually hang out in various lymphoid tissues of the body for the next exposure with the same antigen in the future. Okay, for memory T cells, it is actually activated. It's almost the same as the formation of memory B cells. Now, activated T cell proliferate to give rise to new T cells. The newly formed T cells, together with the originally activated T cell, form the memory T cell, which also hang out, in various lymphoid tissues of the body, just like the memory B cells too. Okay, now the primary and secondary responses. The primary response is the first contact of B cells with antigen, which is slow to act, less potent, and transient, or for a short period. It is associated with IgM secretion. All right. Then for secondary response, it's a subsequent exposure to antigen. Okay, that's the second contact. Okay, now it's quick to act or respond. It's very potent and long lasting, that is for many months. Okay, it is associated with IgG secretion. All right, now let's look at the classes of antibodies. We have 5 major classes of antibodies. We have IgG, A, M, E, D, which you can acronymize as GAMED. Okay, IgG is involved in complement fixation and also important in secondary response. IgA is present in secretions such as saliva and tears. IgM is involved in primary response, also involved in complement fixation. IgE is involved in allergic and hypersensitive reactions. Then finally, IgD is involved in recognition of antigen by the B lymphocytes. All right, let's look at the mechanism of their actions. Antibody can perform their action in 2 ways: by direct attack on the invading organisms or activation of complement system. Okay, now let's look at the T cells. The T cells involves the helper T cells, the cytotoxic T cells, and the suppressor T cells. The helper T cells are in 2 forms. We have Th1 and Th2. Th1 secretes interleukin 2 and gamma interferon. Th2 secretes interleukin 4 and 5. So Th1 is associated with cell-mediated immunity, while Th2 is associated with humoral immunity. Now, the helper T cells receive antigen from antigen-presenting cell via MHC2 proteins. All right. For cytotoxic T cells, we also have 2 forms too. We have the alpha-beta form and gamma-delta form. They also receive their antigen from antigen-presenting cell via MH1 proteins. Okay, the cytotoxic T cells, they secrete perforins and destroy their target cell. All right. Now, for suppressor T cells are involved in maintaining immune tolerance. Okay, by actually mediating the effect of cytotoxic T cells and helper T cells too. All right. Now, helper T cells plus suppressor T cells are actually called regulatory T cells. Now, we can have what we call active and passive immunity. Now, both are actually form of acquired immunity, but the difference is active immunity is actually the formation of acquired immunity within the person's body, while passive immunity is actually a form of acquired immunity that was got from another body or another human being. Okay, now we have allergy and hypersensitivity. Okay, allergy and hypersensitivity. We have allergic reactions, which are anaphylaxis, urticaria, hay fever, asthma, and rhinitis. Now, allergy is actually excessive response or hypersensitive response of immune cells to antigens, okay, which results in those allergic reactions. All right, now let's look at red blood cell antigenicity. Now, red blood cell antigenicity involves different antigens located on the surface of red blood cell. Okay, we have a number of them. but the two prominent ones are actually the ABO system and the rhesus system. Now for the ABO system, we have majorly the A and B antigens, okay, which actually determines the blood group. That is whether you are blood group A, B, AB and O. Okay. Now we also have the rhesus system. The rhesus system involves the rhesus positive and rhesus negative. Now that's based on rhesus antigens. If someone has rhesus D, that's rhesus capital D antigen, that means you're rhesus positive. So other rhesus antigens are rhesus negative. Okay. Now blood typing and blood matching. Blood typing is actually the determination of somebody's blood group, while blood matching is the determination of compatibility between the donor's blood and recipient's blood. Okay. Now we have the hemolytic diseases of the newborn, or we can say the hemolytic conditions of the newborn. Now it can be in two forms. You can have physiological and pathological. Now physiological is actually changing of the hemoglobin type from fetal hemoglobin to hemoglobin A, actually cause hemolysis of some red blood cells, which we also call the hemolytic condition of the newborn. Then we also have pathological, like in rhesus incompatibility and ABO incompatibility. Now rhesus incompatibility as in erythroblastosis fetalis, that is when there is incompatibility with the rhesus antigens, like when the mother is rhesus negative and the baby or the fetus is rhesus positive, and so there is there's reactions for the subsequent babies. Okay. Now let's also look at tissue and organ transplantation. Now for this, it can be in 4 different forms. You can have isograft, that is iso means the same, so like in identical twins. Okay. We have allograft. Allo means other. Okay. So in maybe the transplantation of tissue or organ from one human being to another human being. Okay. Xenograft means foreign, so transplantation of organ or tissue from animal to human being or vice versa. Then autograft, auto means self, so the transplantation within the person's body. Okay. Now let's look at hemostasis. Okay. Now hemostasis is actually the process of arrest or stopping of bleeding, okay, or the prevention of escape of blood from the blood vessels. Okay. Now hemostasis involves 4 major processes. We have vasoconstriction, we have platelet plug formation, we have blood clotting, and finally we have formation of fibrotic tissue. Now starting with vasoconstriction. Now vasoconstriction involves a number of nervous reflexes, local myogenic contraction, and release of certain corticoids. Okay. Platelet plug formation involves the release of thrombosin A2 and ADP. That is a clustering of the platelets from the blood into the injured blood vessel. All right. Then blood clotting actually involves 3 major processes: the formation of prothrombin activator, formation of prothrombin, and the formation of fibrin. Okay. Now formation of prothrombin activator involves 2 major pathways that we call intrinsic and extrinsic pathways. All right. Intrinsic within the vascular system of blood vessel, extrinsic that is outside the blood vessel. Okay. Now intrinsic involves the exposure to collagen fibers and rough surface of the blood vessel wall, actually cause the activation of factor 12. Okay. So activated factor 12 activate factor 11, activated factor 11 activate factor 9, which actually form complex with factor 8 when it becomes detached from von Willebrand factor. Okay. So factor 8 becomes activated when it detaches from von Willebrand factor. All right. The complex now activate factor 10. Now activated factor 10 in the presence of factor 5, phospholipid, and calcium ions actually form prothrombin activator. All right. Now that is for intrinsic pathway. For extrinsic pathway, we have trauma in the tissue, and the tissue factor actually activates factor 7. The complex activate factor 9, which actually form complex with factor 10. Okay. Now factor 10 in the presence of factor 5 and calcium ions actually cause the formation of prothrombin activator. Okay, please, you need to note that both pathways actually end up forming factor 10, and factor 5 and calcium ion are actually required for formation of prothrombin activator. Alright, so now after the formation of prothrombin activator, then prothrombin activator will now catalyze the formation of prothrombin to thrombin, while thrombin will now cause the formation of fibrin from fibrinogen, okay, which we form fibrin fibers that we mesh up with blood vessel to actually close that injured blood vessel. Alright, then let's look at the clotting factors and their synonyms. Now, fibrinogen is the factor 1, prothrombin is factor 2, tissue factor is factor 3, calcium is factor 4, factor 5 is actually labile factor, factor 7 there's no factor 6, factor 7 is a stable factor, factor 8 is anti-hemophilic factor A, factor 9 is Christmas factor or anti-hemophilic factor B, factor 10 is tract factor, factor 11 is anti-hemophilic factor C, factor 12 is Ageman factor, and factor 13 is actually fibrin stabilizing factor. Now, other clotting factors are prekallikrein, high molecular weight kininogen, and platelets themselves. Okay, now finally we look at the formation of fibrotic tissue. Now, fibrotic tissue is actually formed when the fibroblasts invade the blood clots and actually form different connective tissue fibers, which will furthermore consolidate the injured blood vessel wall. Okay, so that actually ends this section. I hope you've actually enjoyed the presentation. Okay, for more enquiries you can call or text the following numbers: 070-3508-3479 or 080-7691-9529. Now, once again, I am Samuel Owoeye, popularly known as Esopians. So thank you very much for listening, and God bless you. You will succeed.